Thank you. Well, I'm super happy to be here. And I guess excited is the word I'll use to tell you about what may have been the most like harrowing professional experience I've ever had. And so let's get into it. I'm going to tell you today about what to do, or what at least what I did when you don't trust your data anymore. And the subtitle here is Why I Retracted 3 of my papers and what I learned from it, and what I think we all can learn from it. Okay, so to give you a little bit of context about this whole story that I'm going to tell you, I first want to tell you about like my research and what motivates my research and the questions I'm interested in. And it's this: I'm very interested in what makes individuals unique. And so a lot of the focus of my research is to understand the influence of the social environment on individual behavior. And so we know that individuals exist in these social environments. And, if you're a sexual organism, you have to interact with another individual at least once in your life to have fitness. And so the social environment is super important. And some of the major hypotheses suggest that individuals are unique in that they have these consistent individual differences in behavior, because these individual differences in behavior help them navigate this complex social environment. And so to make that a bit more specific, there was a hypothesis that I got really excited about when I was doing my PhD, and it's called the social niche hypothesis. And the general idea here is that in stable social groups, behavioral differentiation among individuals can help reduce conflict, right? And then consistency in that behavior, so if an individual picks a role and sticks with it, can help enable better cooperation, either to do like a cooperative task or to avoid each other if they're doing a competitive task. And so the social niche hypothesis, I think, is cool, it's intuitive, and it makes a very major prediction, which is that these individual differences in behavior should get stronger the longer a group has been together. And so that's what I'm trying to show here with this little schematic where, if you were to measure a group of, say, 4 individuals, every line is an individual, over time, you'd see that these individual differences get bigger, right? And so this is the social niche hypothesis, and I tested it as part of my dissertation. And so my dissertation I did on 3-spine sticklebacks. And so what I did is I manipulated how long the groups had been living together. So I had groups that were very highly familiar and groups that were not very familiar. With the prediction being that the highly familiar groups would show stronger individual differences. And lo and behold, was not the case. It turns out sticklebacks have very strong, personalities, regardless of their familiarity with their group mates. It doesn't matter. And so I was thinking about this, and I'm like, okay, so does this mean the social niche hypothesis is no good at all, or is it maybe just not good in this species? And so I really was thinking about this, and I was like, well, social niches may not really be relevant for species that live in dynamic groups like sticklebacks. They're not in like stable groups or kind of, fission, fusion, always meeting new individuals. And so really, if social niches are going to be important, they're going to be important in species that have very stable social groups. Enter social spiders. And so I was at the end of my PhD at this point, and I went to the ISBE meeting, I think it was in 2012 in Sweden. And I was essentially just fishing around for like postdoc opportunities and collaborative opportunities. And I knew of Jonathan Pruitt. He at the time was, this rising star in behavioral ecology, doing lots of very cool work. And he was working with this really incredible system, which it's incredible, but it's like, if you've never heard of social spiders, they are the thing of nightmares. They live together in these very stable social groups their entire lives. And so they build these massive webs that they use to capture very big prey. They do alloparental care. Everybody can have kids, so it's not like a eusocial insects. They just, cooperate together. And so I approached Jonathan and I basically said, hey, there's this like cool hypothesis that I'm really excited about. And, I don't think it's a thing in sticklebacks, but if it's going to be a thing anywhere, it's going to be in your social spiders. So I said, would you want to collaborate and test this with me? And he was very on board, very excited about it. And so at that conference, we sat down and we designed a bunch of experiments. And so the idea here was that we would measure the behavior of individuals repeatedly, so say 5 times, prior to some sort of treatment. So we get an estimate of what their normal behavior is. And then we would put them into these different treatments where they'd live together with groups for different amounts of time. Then we'd measure their behavior again after they had been in these social groups, with the idea being that individuals that were in groups for only a short amount of time would not have very strong individual differences, right? They haven't settled into their social roles yet. But individuals that were in very highly familiar groups, and have been in this stable group for a long time, they would now show evidence of these social niches if this was happening. And so that's what we designed. And that was in summer of 2012. And so flash forward now, however, 9 months later, March of 2013, Jonathan sends me this email, and here's a little bit of foreshadowing. He tells me, FYI, organizing this data file was a huge pain. Was it, Jonathan? And so he sent me the data file, and I was really excited to get started on the analysis. And so I just want to give you a little bit of context here of what is the data going to look like. And so Jonathan said that they collected data on boldness, which is really just a response to a simulated predator attack. And so what he said they did was that he and his undergrads would put a spider in some sort of dish, and they would puff the spider with some air to simulate a predator attack. And then universally, these spiders, they curl up their legs and they go into this little huddle position. And then he would measure how long it took them to recover and start walking around normally. So that's the type of data we're going to talk about. It is time data in milliseconds, or in seconds and milliseconds. And so I open up the data file, I import it into R, and I start looking at the data. And here's what it looks like. And so the important thing to know is that the trials were only 10 minutes long. And so that's why we have a ton of excess, or that's why he said we have a ton of XX 600 values, because that's the maximum amount of time. So all these spiders were supposedly the ones that never recovered after the simulated predator attack. So I knew that was going to be a problem, but then otherwise, I don't know, this looks like pretty good data. There's tons of variation in it. There's no obvious outliers. There's no obvious patterns when you look at it graphically, which is what people typically do when they get their data files. Okay, so we have our data. It looks good. Let's test our hypothesis. And so remember, our major prediction here is that these spiders that are in more familiar groups are going to show stronger individual differences, which we measure using repeatability. So a high repeatability value just means that there's stronger individual differences. Lower repeatability value means that there's not as strong individual differences. And guess what? That's exactly what we found. And so here is the figure from our first paper. And so the black bars, you can kind of ignore them. Those are control groups that Jonathan said he did. And we expected the repeatability there to be high in all of them. The interesting groups are the gray groups. And so those are our treatment groups. And as you can see is that when spiders have only been living together for 1 week, they don't show very strong individual differences. But once they start living together for 4 weeks and they're highly familiar, they show these really strong individual differences, indicative that they have some sort of social niche. This was so cool. Here I am. I think, I am at the end of my PhD. I'm so excited about these results. I'm this is just the coolest thing in the world. The paper was a breeze to write because, the predictions were so clear, the data were so clear. And so we published our first paper in 2014 in Proc B. We then published another one in a different species, supposedly finding the exact same thing. Then we published a third paper where we actually showed that these social roles, these social niches supposedly had positive fitness consequences for the individuals. So that was great, right? And so for the rest I'll end up referring to these papers a lot. So I just want to highlight that I'm going to talk about, I'll call them the Proc B paper, and I'll call them the AMNAT paper. So just keep that in your head. That's what I'm talking about here. Okay. That was fun. That was this great collaboration. I did it right at the end of my PhD, at the beginning of my first postdoc. I was really proud of myself for, establishing this independent collaboration with this, kind of a big deal professor, but it was a side project for me. And so, I did my postdoc in Germany. I work on fish. I kept working on fish. And so now flash forward. So those papers were published in 2014 and 2016. And so now flash forward, about 5 years to 2019. It is fall 2019, and I've just started, my dream job as a faculty at UC Davis. I mean, getting a tenure track position for those of us that want to stay in academia is like such a big deal. And I was like, just like on cloud nine, so happy to be at this school and like back in the States. And then I get this. So I get this email on November, go back, on whatever that date is, November 8th of 2019. So I've only been a faculty for 2 months. And I got this email from someone I know and someone I trust and I consider a mentor of mine. And they sent me this email that says, "Hi, Kate. There's a somewhat delicate confidential issue where I would value your opinion." And I was like, "Oh, okay. I don't know. Are you going to like nominate me for an award or something?" And that is that's not what it was. And so this colleague, we Zoom. And so this colleague says, "So funny thing is I downloaded the publicly available data for your AMNAT paper." So this is the 3rd paper in the series. "For your AMNAT paper. And I noticed some really weird things. It seemed like there's a lot of duplicate values." And I'm like, okay, well, we know that like there's going to be a ton of 600 numbers because that was like the maximum time for the trial. So those are expected. And he's like, no, I don't mean the 600s. I mean that there's exact duplicates of your behavioral trial values across a lot of spiders. And so I'm like, okay. And so here's what the data looked like that was deposited on Dryad as part of the AMNAT. And so you can see there's like identifying columns, a source colony, their experimental colony. Here's the individual ID. And so this is what we call like the long format, where every row is an observation. And so every individual has 5 rows. I hope that makes sense. And you can see that here's their boldness score before their treatment, and here's their boldness score after the treatment. And so I'm looking at it and I'm like, well, okay, so where are these duplicates? So what I do is I sort it in ascending value. And lo and behold, there are these. And they're like all over. And so this means that 3 different spiders all recovered at 0.23 seconds. Or like these 2 spiders recovered at 3.91 seconds. And I'm like, that's weird. Like, why on earth would they be recovering like at the exact same millisecond? And so my first thought, my heart drops and I'm just like, oh God, what is happening? And my first hypothesis is that I'm an idiot and I somehow messed up the original file during analysis. Totally possible, right? Luckily, I started grad school working in menu-driven SPSS for data analysis, but then by the time I ended grad school, I had switched over to R, which meant that I had the scripts of everything that I did to these data. And so here are my scripts from this group FAM analysis is what I called it. And you can see everything I did. And so I was able to rerun my code with the original data file. And I could say, no, I did not do this. These duplicate values are present in the original data file and nothing I did has altered that. So I can reject this hypothesis. So then I'm like, well, I still don't know why these things are duplicated. Based on my understanding of the experimental design, this shouldn't, like, how on earth could you measure like 2 different spiders and get like the exact same value so many times? So I went to Jonathan and I, of course, said, Jonathan, you collected these data. Like, what's going on here? I don't understand why we would get so many duplicate values. And so my understanding of the experimental design, as he explained it to me, was that they had a spider in a dish, they scared it, and then they started a stopwatch and they measured how long it took them to recover. And he says, no, that's not what we did. Instead, we measured blocks of several dozen spiders simultaneously. And so the observers do not stop the timers when a spider moves because other spiders are being measured using the same timer. And so he's saying basically now that as opposed to, a single spider, they have like multiple dishes with multiple spiders. They scare all of them and then they start one timer. And so if like 2 spiders, start recovering at about the same time, they'll just look at the timer and say like, oh, it recovered at 3.91 seconds. And so I was like, man, So I was a little annoyed and honestly a little embarrassed because this meant that I had published a paper where I did not fully understand the methods. And so I wasn't crazy about this. Jonathan wanted to publish a correction that just says, oh, here's the new methods. And I was like, well, like this means now that the spiders are basically being run in blocks, right? And so this means that these values are not independent. And so we need to account for this in our statistics. And so I asked Jonathan, I was like, can you send me the schedule of how you ran these spiders in blocks? So that way I can include that as a random effect in our statistical models. So if we correct this paper, at least we can update the analyses too and see if our results still hold. However, he says no such schedule exists, or if it did, it's been lost. And so I was frustrated with this. I didn't want to publish a correction that was kind of like, didn't have the appropriate statistics now done on it. And I was like, well, I bet I can recreate this by hand. And so what do I mean? So the idea here is that if spiders are getting the same value because they're being run in these blocks, then we should see the duplicates occur in the same observation period. And so here's the same data I showed you before, but now this is in the original wide format that Jonathan emailed to me. And so now, as opposed to every row being an observation, every row is an individual. So here's this individual. There are 5 boldness scores before the treatment. I just added this gray bar to make it more obvious this is where the treatment happened. And then there are 5 boldness scores after the treatment. Okay, so you all are going on my deep dive with me for my crazy Excel files. I've looked at so many Excel files for so many hours. And so my hypothesis was, well, if they're being, all the duplicate values now should be in the same observation, right? Because they're being run in these blocks. And so if I can figure out where the duplicates are, those spiders I could assign to the same block, and then somehow I'll be able to figure out, basically have a proxy for what block they were in, and I could add that in now as a random effect when we publish our correction. And so I just start control Fing in this Excel file, looking for these duplicate values, expecting them to be in the same column. They are not. And so here's what you're seeing here. And so this value right here, 257.62, 257, the duplicate value is not in the same column. It's right here in the same exact animal, separated by supposedly a 5-week familiarity treatment. So here's 257.62. Here's 124.72, and here's 124.72. So every value here in red is duplicated across the familiarity treatment in the same animals. So I can safely reject this block design, right? These duplicates are not occurring in the same observation period. They're occurring in the same animal. And so this explanation that, someone started a stopwatch and just didn't stop it and gave the same value to different animals, it's just not true. So next hypothesis is I was thinking, okay, so now what we're basically seeing is we're seeing duplication across columns. And so if these cross-treatment duplications are a result of copy-paste errors, then we should see entire blocks of the same number. So I don't know about you, have you ever done the thing in Excel where you highlight a column, and then right at the bottom of the column, if you highlight it, there'll be a little plus sign. And if you grab that plus sign and pull it over, you will duplicate the whole thing. And so this has happened. I've done it by accident before. And that was my next hypothesis as well. Maybe that's what happened. Jonathan accidentally highlighted some rows and just pulled them over. And now that's why the duplications exist. And so I just kept color coding these data files, looking for that pattern to see if what was going on. And so here's some examples of what this looks like now. And so now the values in red are exactly duplicated across this 5-week treatment that he supposedly applied. The values in yellow are almost duplicated, but not quite. So this value here, 77.56, 477.56. This value here, 212.22, 412.22. And so this is weird, right? These values are almost identical, but not quite. And it's not even they're different in the same direction. So this one's 400 higher, this one's 200 lower, or 200 higher, this one's 200 lower. And so this is just some of the examples of the color coding that I did. You could color code this whole file and you see this pattern across the entire thing. So the bottom line is I can reject this hypothesis too. And so there's no, as far as I can tell, there is no methodological explanation for this. There's no biological explanation for this. And so what did I do? I retracted the paper. And so I asked Jonathan, I said, I don't trust this data anymore. I don't know. Something happened to this data file. He kept saying the data file got corrupted. And I was like, you know what? Like, doesn't matter what happened. I don't trust the data. I don't trust the results. We need to retract this. And so we did. And so I tweeted about this in early January 2020. I'm starting the year off with something I didn't expect to ever do. I'm retracting a paper. And I recently discovered major problems in the raw data associated with it, and so the results shouldn't be trusted. And so that's not the end of the story. And so this was the AMNAP paper. This is the AMNAP paper. This is paper 3 out of, so this is the latest paper. But, you know, there are other papers. There are 2 other papers in this series that I did that Jonathan collected data for me on. And so I said, well, let me look back at these other papers. And so now I'm going to look at the Proc B paper. And so this is the first paper in the series. And what did I do? I opened up the data file. Again, here it's in the long format. I sorted it, and now I've color coded it. Ooh, and there are duplicate values down to the millisecond again. And so this is a problem. And then I did the same exact thing that I did previously is as opposed to looking at the long format, which was deposited, I opened up the original data file that Jonathan sent me, which was in this wide format. And so now every row is an individual. So here's like a snippet of like individuals that are in the 3-week familiarity treatment. And here's a snippet of individuals that are in the 4-week familiarity treatment. And guess what? This whole block of red is identical in 2 different treatments. So like 600, 484, 75, 600, 484, 75. The whole damn thing. And I think it's really curious that these duplications, these blocks of duplicated numbers are even more obvious in this earlier paper, right? We don't have any of these yellow values, which is what we had in the AMNAP paper. And so at this point, I emailed Jonathan. I candidly asked him, I'm like, what's going on? Why do these data look like this? And his response, this is an interesting question. It sure is, right? And I don't know the answer. Well, I know my answer to it, but he never gave me an answer. So what did I do? I retracted that paper. And so I'm not excited to say that there were similar problems in the raw data of another of my papers. This paper is now retracted. And then I did the thing that like, this is when all hell broke loose, right? Is that I know there might be some interest in what's going on here. So here's the full account of how and why I came to this decision. And I published this blog post on my lab website that basically went through everything I just told you and then in far more detail. And there's other things going on too. So if you're interested in the full story, go read that. And I don't know, I published this blog post and I was basically thinking like, I have to somehow, you know, show what is happening and like what I'm seeing. And people ask me why I published that blog post. And really this was my thought process is like mistakes happen, you know, maybe I didn't explore the data as well as I should have. I don't know. But they can be corrected, right? It's easy to make mistakes. We're all human. Shit happens. But I'd rather be considered an honest idiot and have everyone wonder if I was untrustworthy. And so this was the thing that really got it for me is I knew that I could publish those retractions and just say like, oh, there's data anomalies, there's data irregularities, and be a little bit like cagey about what was going on. And that's certainly what Jonathan thought I was going to do. However, one retraction for like data anomalies is like, oh, you know, whatever, that's kind of a weird thing that happened. 2 retractions for data anomalies, and then 3, I knew that every conference I went to from there on out, everyone would be like, what's up with Kate? Why did she retract those 3 papers? That's so weird. And so I figured the only way for me to get out of this was to just be completely honest, completely transparent and say, this is what I'm seeing. Here are all the hypotheses I tested. What do you all think is going on? And did I make the right move? And so I hope I made the right move. I think I have. But everyone always asks me what happens next, what happened next. And so I like to put this timeline on there because just please, like, no good deed goes unpunished. And so I published my first retraction on January 17th of 2020. I published a second one and my blog post on January 29th of 2020. And then very quickly, lots of Jonathan's other co-authors contacted me, right? And so this apparently was something Jonathan would do, is he would either approach other early career researchers or they would approach him with ideas. He would use his army of undergrads to collect data and then send them the data, and they'd write up these papers, right? It seemed like a great collaborative effort that, you know, he was involved with all these other early career researchers. And so a lot of these people, I knew them personally, and so they contacted me and I just said, look at your data. And so they found all sorts of problems similar to mine, some other things as well. And so shortly after this, when this clearly becomes evident to be not just 2 retractions, but a much bigger deal, Pruitt gets lawyers, as is his right, as well he should. And then a really interesting thing happened. My impression is that when he realized what a big deal this was going to be, is he actually tried to unretract my papers. And he says this. And the reason why is, he said, is that Dr. Laskowski's statements have misled the public and my peers. She refuses to clarify the matter now, which implies malice and intent. It is clear now that the retraction statement and the publications of Dr. Laskowski led me astray, and they continue to misrepresent the facts to the public. So this was stressful because this is obviously legal language. And so I was very much felt like I was being threatened with lawsuits or potential lawsuits. And that was stressful to say the least. And then in November of 2020, I was contacted by the investigative team. At this point, Jonathan was at McMaster University in Canada. And so I started working with them, providing them all the evidence I've shown you. I had lots of depositions with the investigative team all across 2021. In fact, when they sent me my final dossier of evidence, including emails and all the data forensics I did, it was 292 pages long. So if you want to know how starting off as an early career faculty was, it was a lot of writing. I can say that for sure. Papers keep getting retracted. It's now up to 15, and there's many more that are under suspicion. In 2022, I am finally told I need to testify in Jonathan's termination hearing because what happened was the investigative team found had their findings, and Jonathan was contesting these findings, as is his right as a tenured faculty. And so I generally freak out now because I do not like the idea of taking the witness stand and getting cross-examined by Jonathan's lawyers. I was really stressed out about this. And the termination hearing was going to be on June 29th, which I know because that's my birthday. And so that sucked. But luckily, sometime before the termination hearing happened, this happened. Behavioral College Jonathan Pruitt has resigned from McMaster. And so McMaster and Jonathan reached some sort of settlement. I don't know the details of what that settlement is, other than this. This was the email they sent me. As part of the settlement, Dr. Pruitt agrees that they will not initiate any legal action against you for making complaints to McMaster University. Thanks. And so it used to be, I thought that's where the story ended. And for well over a year, this was the ending slide for this until May 10th of 2023, when this happened. On a random Wednesday, 3.5 years later, the craziest chapter of my career comes to a close. McMaster released summarized findings of their Pruitt data investigations. The committee found that Dr. Pruitt engaged in fabrication. And here's that summary that they sent me, and you can see the important bit that's highlighted, that Dr. Pruitt engaged in fabrication and falsification. And so I hope this is the end of this story. I don't know what else could happen. I hope nothing else. But yeah. And so what have I learned from this whole experience? What can I hope that we can learn collectively as a field? But first and foremost is, man, keep a paper trail. This is why I think Sortie is so great, because they're pushing for all of these policies and to really, make this cultural change. Keep notebooks, keep schedules of your experimental design, take pictures, take videos, get really good at doing version control of your data and your code so it can be completely reproducible. Use a scripting language so that anyone else can reproduce your analyses. And I'll also say, I really think that open science is necessary. This was really only discovered because the data sets were publicly available and someone went digging through them. And so deposit your data, deposit your code. And then, I mean, truly the only reason I think I, quote unquote, survived with my reputation intact, at least as far as I can tell, is because I took, I was like, the only way I can do this is if I'm completely honest and completely transparent and completely public. And so that was the tact I took, and it seems to have worked out. And the other thing I think is important is, like, this whole thing sucked, right? Like, I would not wish this on almost anyone. But I do appreciate and am glad that it does seem to have now been this learning moment, and actually a really positive teachable moment in some ways. And so I have received some of the nicest emails from people across the community as a result of this. And I think this is the one that just gives me hope, man. This one in purple, where they wrote me this really nice email and they said, I have never been more proud to be a scientist. It gives me hope there are scientists and professors out there fighting to do what is right. And so that is what we're doing. So then, I know I went a little bit over, I'm sorry. But then the last thing I just always like to say is, I'm an animal behaviorist, and so that's why I have very pretty pictures of animals right here. But please imagine whatever thing gets you motivated if you're not an animal behavior person. But I just want to say that all of us are in this job because we're fascinated by the natural world. And for me, particularly by the animals that we inhabit this world with. We want to understand how and why these creatures do the things that they do. And the only problem, though, is that we can't directly ask them this, right? They're not humans. They can't tell us why they do the things they do. But they can communicate with us. And they do so through the data that we collect on them. And so I want to say here that the integrity of the data we collect is paramount. Whatever story the data tells us, we need to believe it and trust it and protect it, even if we don't necessarily like what it says. Manage your data carefully along every step of the process. Be a faithful translator of what the animals tell us, and don't be tempted to tell the animal's story for them. Their data is their voice, so let them speak for themselves. And so with that, I will end this and take any questions.
Awesome, Kate. Thank you so much. It's been a roller coaster ever since the beginning, emotions, everything that you went through. And I also really admire you. So we will start a general discussion. I think we have between 10 and 15 minutes for questions. We encourage all the participants to use raise hand. And Kate, maybe you can then call their names. And also we have several questions in the chat. So whatever works best for you.
Well, yeah, I see Kurt saying, did this all snowshoot from a spreadsheet gone wrong? Yeah, I think that's one way to put it. It was not an accident, right? This wasn't some data corruption, as you can see. And then Susanna says, I'm curious, how long did it take to retract the first paper, and was the process easy? So let's see, I got that email about the first paper, the AmNat paper, in November. And then the retraction, happened in January. And so that included, I mean, God, there was probably 2 solid months of just intense, on my part, deep diving data forensics, lots of back and forth with Jonathan. He would come up with new explanations, and I would, basically test them in the data and could, reject them all. So in that case, the retraction took about 2 months, and Jonathan was on board with it. He approved the language in the retraction statement. And so those first 2 retractions of mine were comparatively easy to do because Jonathan agreed to everything. And so that process was relatively easy, especially because the editor at AmNat is Dan Bulnick, who at the time, who himself had retracted a paper because of an R code mistake. And so he was very understanding and helpful. After that, the process got significantly less easy as Jonathan, you know, got lawyers and then fought almost every single retraction after that, is my impression, and did did his best to really slow down the process. And I now have very strong opinions on the editorial teams at several of our journals. Some of the editors I think were very helpful and adjudicated this process well. Other editors were very intimidated by lawyers and clearly were only seeking to protect the legal threat and not necessarily the scientific record.
I think we had one hand raised. So was it Julia or someone?
Yeah, so my question was the same as the one in the chat, which was just how easy the retraction process was. Because I feel like that's not something that's necessarily discussed super openly, especially in early career, of like how you even go about that. Yeah, that was just really insightful answer.
Yeah, I mean, the first step is contact the editor, and then retractions are comparatively easy if everyone agrees to them, all the authors. But when all the authors don't agree, can obviously be the case, especially when there's any sort of allegations of misconduct. That is where things get very highly variable in my experience. Yeah. Yeah, I just hope everyone can learn from this. Like, please, like, keep track of, like, use version control on your data and like keep track of all these things. That has definitely been that's something we now do in our lab. We're really good about this, but documenting everything you do is like really, really important. Yeah, Lena.
Hi,
Hello.
can you hear me?
Yep.
Perfect. Thanks very much for this really, really interesting talk. My question is, I'm trying to think of all those filters and reviewing stages, official and unofficial, that those papers went through until they got published. Could you briefly like describe those filters? Like how many people missed that? I'm not sure if they really missed it, because if you're not looking for such stuff, you're not gonna find them. But could you please briefly explain what the stages and the process that those results went through?
Yeah, I mean, I think this is a really good point. So, I mean, these data and these papers went through every stage of the process. So, like, the data first got sent to me. And like I showed you some of the data exploration I did, but like I consider myself kind of a statistical expert. Like, I teach a graduate level course on statistical modeling. So it's not like I was some dodo who just like got this data file and just like willy-nilly started plugging and chugging numbers. Like, I did proper data exploration. I graphed everything. I looked through the Excel sheets. I made sure that nothing seemed out of the ordinary, and nothing did seem out of the ordinary. The only thing I didn't do, because it never even crossed my mind that this would be a thing, is I never sorted my data to look for duplicate values. I graphed it, but of course, then when you graph it, you can't see the duplicate values because they're right on top of each other. So I think in this case, these problems were designed not to be easily discovered, and they were not easily discovered. And so I think in this case, I don't know that I think all the steps of the process worked the way they were supposed to. That being said, I think all the steps of the process in terms of good data exploration, double checking your data, having your code and rerunning your code cleanly before you submit anything, those are all really, really good steps, because I think the vast majority of problems with data are truly just like honest mistakes, right? Like, you enter something wrong, you accidentally copy paste something. And I think that is where those steps are going to help us the most, is like in catching our own honest mistakes. I mean, the problem is bad actors are bad actors, and any rules you put in place, they will find a way around them. And so there's no system we can create that's perfect, but we can do our best to, catch the honest mistakes and make it really annoyingly difficult to try to be a bad actor. Okay, let's see. Sergio has a question. what are your thoughts on trust after your experience? What's your approach to starting new collaborations where you did not collect the data? I mean, everyone always asks me this if like I'm now jaded, and I hope the answer is no. I still trust my collaborators. That being said, I haven't really had a big, other than my own students who I mean, I guess I would consider them my collaborators. Yeah, I don't know that I've had a collaboration where someone at a completely different lab has collected the data without me ever seeing anything. So that I would be a little bit nervous about. I would certainly ask for, photos and videos, not because I didn't necessarily trust them, but just because I think, I'd want to catch if like, oh, this experimental design doesn't seem quite right, or like, I still trust them, but I do think there's value in good documentation, mainly because it catches mistakes. And so now, like with my lab, I mean, they all went through this experience sort of with me. And so they know, like, and I've told them, like, we live in a glass house. Everything we do in my lab is going to be, is publicly available. We deposit our data, we deposit our code. They have to upload photos of their lab notebook every month. I rerun their code oftentimes. And so I think those are just all the steps you can take, is like, you just put in layers of protection to catch mistakes, and also then layers of protection to make it really annoying in case someone did want to act in bad faith. Yeah. Tim, did you want to?
I mean, if you want to answer one of the other questions in the chat that came up before me, I think that's fine.
Oh, yeah. So Sarah says, do you have any thoughts on what else editors, journals can do in addition to asking authors to submit their data? I actually think, definitely submit data, absolutely. Submit code, but I would almost say, in addition, I think the R markdown files are more useful in a sense, because, if you submit your code and your data, that means you're asking the editor or reviewer to rerun your code. Let's be honest, very few people have time for. But if you submit the R markdown, at least then we can see how you run your code. Ideally, of course, reviewers will run the code to make sure it's reproducible on a different system, different machine. But I think running that R markdown helps a lot. And it also helps in terms of like us learning from each other. And I know some journals are also moving into having like actual data editors. I know AmNet has done this. And so they have an editor now that goes through all of the data and code and metadata to make sure it looks like reproducible and has like good metadata. So I think that's, what we need to, that's sort of the method we need to go in, but that does require like, systematic support of now having a new editorial role for that. Okay, and then Anita, yeah. Has this situation affected you in your career or with your institution? I think not negatively, surprisingly. So when I first got that email, or when I first Zoomed with my colleague and they made me aware that there was problems, and once I got the first hunch that this was more than just like a little honest mistake, I actually contacted my PhD supervisor, Allison Bell, and I was like, oh my God, Allison, like, what do I do? And she immediately told me, like, go talk to your chair. And so I went to my brand new department chair. I'd only known him for 3 months. And I said, hey, here's this thing that's happening. And funny thing, Jonathan Pruitt was a postdoc here at Davis. And so people knew him here. And so Jay, my chair at the time, was super supportive. He gave me access to UC Davis lawyers. They blanket believed me from the get-go, which was really nice. So I think in that sense, they like UC Davis has been nothing but supportive. They've given me access to all the resources. And so I was very honest. The legal threats were the most terrifying thing, because obviously that could be lots of money. But they gave me access to lawyers, and it actually made me feel really good when I sent those statements that I shared with Pruitt, where he said, I'm acting with malice and intent. I sent those to the UC Davis lawyers, and they were like, oh yeah, he's trying to build a defamation case. And the lawyer was like, I'd like to see him try. And she just laughed and laughed. That made me actually feel like that was the moment where I was like, okay, like this will be fine. I could survive this. So if anything, I don't know if I've benefited from this. I guess now here I'm talking to you all because I did this. So it hasn't negatively affected me, which is a big relief. Yeah.
Okay, maybe Tim, because he's waiting. And then we have a question from Leonardo in chat.
Sure.
Thanks, Maria and Kate. Yeah, so I guess you touched on what I'm about to ask a little bit, but I just want to ask it a little bit more explicitly. I think maybe, I don't know if you're familiar with some papers that were published some years ago now, well, doesn't really matter by whom, but papers that made the case that all research, everything that we're doing should always be videotaped to demonstrate that we're not making stuff up, basically. And I guess I wonder what your perspective on that argument is, that we need to just essentially raise the bar of documentation across the board, everywhere, to ward off these threats of fraud. I mean, you sort of addressed this already, but I just wanted to specifically mention that.
I mean, that seems pretty extreme, right? I mean, that's not a trivial thing to videotape week-long behavioral experiments. And so funnily enough, when this whole thing was going down all through 2020 and we were in lockdown, and Jonathan was fighting back against all these retractions, and what he was in, I think he was in Florida at the time. And so what he did is he wore a GoPro and he was videotaping himself collecting data on spiders to prove that these data were real, or that the results at least were replicable. I mean, I do think we need to raise the bar on documentation. Maybe videotaping is the way to do it. All my behavioral, like I study fish behavior and development of behavior, and everything we do is videotape. Like that's how we make our money. But honestly, like we produce terabytes of videos a month. And so I just don't know if that's like feasible. I think documentation in terms of code, data, pictures, and maybe for some key things, videos are important, but as a blanket statement, I don't know, that feels a little bit maybe too extreme. That's my gut feeling, but I could be convinced otherwise. And then Leonardo asks, does the hypothesis you tested in the retracted papers, has it been tested or approved from other researchers since 2014? Funny. And so I actually did one of my pandemic projects is I got asked to write a review for the annual review of ecology, evolution, and systematics. And so as a response to basically this of like since now all these retractions have happened, like what do we have evidence for? And so I encourage you to look up this paper, Laskowski et al. 2022. And the most important thing is that in that paper, we have what we call the supplemental table of shame, where every paper that was retracted, we have a table of like what that hypothesis the paper was supposedly testing, what the retracted paper found, and then other papers that have also tested that hypothesis. And as far as I can tell, several other groups have tested the social niche hypothesis in very different systems, in like chickens and meerkats, mice sticklebacks. None have found support for it. But no one's tried to retest it in the social spiders. I mean, there's other people who work on social spiders and no one has retested it yet, to my knowledge. I hope they do. I mean, I think it would be very cool to do.
So Kate, I have a question if I may jump in. So I just typed in the chat that we have around 5 or 6 minutes more. So if anyone has any thoughts or questions, just raise your hand or type in the chat. My question would be, do you see Sortie helping with this situation or similar situations, or what could be our role in helping with more transparent work and also helping with similar cases like you had?
I mean, yeah, I mean, I think you guys are doing God's work in terms of, advocating for this better documentation. I think that's like step one, right? Like create a culture where things are much more better documented, both in terms of like data deposition should be by far the rule. I mean, of course, there's exceptions if you work on humans or something like this, but I think generally it should be the rule. Data code deposition should be the rule. And just other sorts of like field specific and field appropriate types of documentation, maybe more videos. I think that's like step one. Training people then also in like better data management is really important. I mean, I don't know if you've ever done this, and I recommend you do it. But this is what I had to do is I had to open up my files, like my folders from 5 years ago, right? And try to figure out what were the important files, what were the final files, like where everything was. And I definitely didn't have as good data file management 5 years ago as I do now. But I think just training in how to do proper file management is really important. How to do proper version control is really important. I mean, you can use Git and GitHub for like code version control. There's also like baby, I call them like data management light, where you never control save anything. You always save as and append the date at the end, and that will give you like all of your data files as you update them, or code files or whatever. So I think that advocacy is really important. And I mean, I don't know if you guys have a role then of like, if other folks go through something similar where they have a co-author that they think is, maybe not using the best research practices, if there's some, I mean, some sort of support group we can offer for them. I've had lots of people reach out to me as a result of this, and I'm always happy to, offer, a sympathetic ear and as much advice as I can. But yeah, I think the cultural shift that Sorty's trying to push is like absolutely the right thing to do.
Awesome. Thank you, Kate. So we had an idea of having breakout rooms where we have 4 to 5 people so we can reflect on your talk.